Okay, good.
Uh, yes.
Okay, good. Good afternoon. If you are in Europe, good morning if you are in the States, and good evening if you are in China. What I'm going to do today is really an update of the webinar that I gave a few months ago. In fact, probably you remember this is the same title, but I think we have to modify the title now because there is no longer a question mark to say that the targeting pancreatic cancer is a reality. Now it's not a question mark. It is a reality, as I hope you will agree with me, when we finish this webinar. Um, just a very quick introduction. The fact that the RAS genes are mutated in one-third of all human tumors. It is the most frequently mutated oncogene, um, with about 20 to 25% of all human tumors. But most important is the fact where KRAS is mutated. It is mutated in the big three, in terms of limited survival. It is in most pancreatic ductal adenocarcinoma, in about half of colorectal carcinomas, and in about a quarter of non-small cell lung carcinomas. So clearly, targeting KRAS is a real emergency or a need, in the world of cancer. Now, just a very brief landmarks, because KRAS is an old friend. KRAS really came to life in 1967 by the hand of Werner Kirsten. He isolated a strain of rat sarcoma virus, and that rat sarcoma virus contained what we now know is the Kirsten RAS oncogene. It's not exactly the same as the one present in human cancer, but is extremely similar, basically the same gene, obviously. So the next important event in this history of KRAS actually came from Channing Dare, because he, in June of '82, published a paper in which he identified an oncogene related to the HRAS oncogene that we and Bob Weinberg and Mike Wigler had isolated described and isolated a couple of months earlier. Then there was a time in which people questioned whether the fact that oncogenes were present in cell lines that have been maintaining culture for many years, we decided to go and try to identify these oncogenes in solid tumors, in tumor biopsies or in surgically removed tumors. And, oops, okay. And we published this paper at the end of 1982 in Nature, in which we described that KRAS oncogenes were frequently identified in human solid tumors. Our main contribution to define KRAS as a true oncogene came about a year later, in February of 1984, when we published, I believe was in Science, that thanks to some tumor we received from Istituto di Tumori in Italy, we showed that even before sequencing was, a routine procedure, that the mutation that activated KRAS was present only in the tumor tissue, but not in normal tissue of the same patient that they sent it to us. And then the relevance of KRAS mutations, KRAS oncogenes in pancreatic cancer came from the hand of a very good friend of mine, Manuel Perucho, another Spaniard, that published in Cancer Cell that KRAS oncogenes were frequently, very frequently, as a matter of fact, activated in pancreatic cancer patients. Then, just to, not make this too long, it is what was absolutely crucial, the development of mouse tumor models. And this has to be given credit to Dave Tubison and Tyler Jacks, that 2001 they published the lung model, and 2003 they published the pancreatic model. Then came what really is going to be the main subject of today and of the coming years, and is the fact that KRAS was thought to be undruggable until Kevan Shokat, working at University of San Francisco, identified a small groove that allow the design the first inhibitors. So, 30 years after the discovery in human tumors, Kevin demonstrated that KRAS oncoproteins were no longer undruggable. Okay. And then, last but not least in this fast, overview about the 60 years of history, we have in 2021 and 2022 the approval of the first two KRAS inhibitors that were selective for the G12C mutation for reasons I'll show you in a second. The designation of the scientific name is sotorasib and adagrasib here. In fact, since then, since 2013, as this nice timeline shows, there have been a series of landmarks in the discovery of different types of KRAS inhibitors. The first, you can see compound 12, it was the original isolate from the Kevan Shokat's lab. And then came the approval, as I just mentioned, sotorasib. Then it came the first non-covalent inhibitor from Mirati, from a biotech company now part of BMS, Bristol Myers, and then Protax and the KRAS inhibitors that were no longer selective for a mutation, that they were pan-KRAS, and then the KRAS on inhibitors or RAS on inhibitors that we are going to discuss today. Indeed, today I'm not going to talk about the Protax. I'm going to talk about these two inhibitors, the sotorasib, adagrasib, and the RMC 6236. Okay, here is sotorasib with the little, with the little groove here in, targeting the switch to region that KRAS uses to engage with the downstream effectors. And this groove is so small that it requires the formation of a covalent bond with cysteine, and that is why the first KRAS inhibitors were only selective for the G12C mutation. This is now sotorasib. This is the overview of adagrasib. Now, let's go to clinical trials. Now, here is the first report of clinical trial with sotorasib in lung cancer. As you can see, there are only a very few cases in which there is complete regressions. So this was a little bit, it was a good news that finally there were selective inhibitors against KRAS, but it was a little bit of disappointment because the therapeutic activity was not what we all expected, okay? So it's as I say here, it's a major advance, but as single agents, their therapeutic efficacy is still limited due to the rapid appearance of tumor resistance, as we will see in a moment. In the case of adagrasib, we have the same story, only basically the waterfall plots are basically identical, and here show you that the 50% progression-free survival is only 6 months, and the 50% overall survival is just a year. Okay. In fact, there is a paper by a colleague of mine here in Madrid, Luis Pazares, reported in Lancet that in fact there is no increased survival using sotorasib versus classical chemotherapy, with the only advantage of sotorasib that the secondary effects, the toxicity, is by far less pronounced than the toxicity due to chemotherapy. But this is really the only advantage in pre-treated patients, obviously. Um, so the question here is why? Why sotorasib, adagrasib, or other inhibitors of the G12C mutant, they're not more effective. Okay, so one could postulate two possibilities. That maybe KRAS is not the only driver, so you inhibit KRAS, but the other drivers keep, pushing the tumor on, or that the inhibitors induce tumor resistance. So to try to give you a picture of what we think is happening, I'm going to take you first to our own work in mouse models, which obviously are easier to manipulate and to generate tumors. This was published a couple of years ago in the Journal of Clinical Investigation. So what we did was to use the tumor model developed by Tyler Jacks, as I mentioned before, and treat these mouse tumors with sotorasib. In mice, this model is not perfect. I mean, assuming that models can be perfect, um, because in mice you cannot, unlike as you will see later in pancreas, in lung, it's almost impossible to generate a mouse with a single tumor or with a couple of tumors, as obviously happens in humans. So you generate, even if you decrease the amount of adenovirus to activate the point mutation, really, per month, per mouse, you always get 5, 10 tumors. So, it's not as clean as we would like to. But anyway, we treated the animals, you can see here, uh, maybe, I don't remember, but maybe 10 mice, total of 50 tumors. And as you can see, the curve, you remember in your retina, the waterfall plot that I showed you a minute ago is very similar, with the difference that there are more complete regressions. But these, as you can see by the color, these are in very tiny tumors. Okay, so obviously in humans the tumors are much bigger. So you just have to look at the middle side of the waterfall plot, and it's really pretty much the same as in humans. So what happens is when you start treating, the tumors regress, but then some just 10 weeks, others take 20 or 25 weeks, they start to grow again. And here you have a picture, uh, a tag picture of the tumor. 4 weeks is less than half of what it was at the beginning. There must be one of those few cases where we were able to get just, one tumor. And then by 20 weeks, now the tumor is double of what it used to be. So clearly the tumor respond the same as in human, but then they become resistant. So this is with the drug, but we decided to ask what happens if we genetically ablate the oncogene, eliminate the oncogene. Do the tumors continue growing, or once they don't have KRAS anymore, the tumor disappear? And in fact, this is what happens. The tumors disappear. You can see here at 2 months, 84% of the tumors completely regress. At 6 months, that figure is grown up to 96%. Okay, basic, and the other 2 or 3 tumors, I think there are just 2 tumors, are just 90% regress. You can see here one red line going up, and it was we're curious to know what was going on in this mouse, because the tumor was growing rather fast, and in fact the animal die, just after 2 months. And it just happened that this was a mutation, a spontaneous mutation in the normal KRAS allele, in the wild type non-targeted KRAS allele. It was a mutation in codon 61. So clearly, the only tumor that grew was because it had a KRAS oncogene, although it was not the one that we designed. So clearly, this showed that at least in mice, KRAS is absolutely essential for tumor growth. And if we eliminate KRAS, there is no resistance. Okay? So now, obviously, the idea will be to either generate a very, very potent KRAS inhibitor in such a way that there will be no longer KRAS signaling, or obviously to go up and try to identify combinatorial therapies. Okay. So this is not just in mice. In fact, there are very two classical papers in humans. One of them came from the laboratory of Andy Aguirre, and published in the New England in 2021. And here is a famous, I mean, I've seen this in many meetings, and I'm sure you too, which represent the various mutations that they identify mostly in liquid biopsies, okay? And the same came from the lab of Pirolito. Uh, um, it's basically the same, a bunch of mutations, but the none of I mean, the important thing here is that KRAS is not being taken over by these mutations. KRAS mutant oncogene is still there. It's not as what happened in other tumor types like the lung tumors with EGFR mutations, or in the CML with BCR-ABL mutations, that when there is a mutation that no longer responds to the drug, that mutation takes over. And this happens, in fact, in those cases in which there is a secondary mutation in KRAS. The drug is no longer effective, there is resistance. But when they have mutations in tyrosine kinase receptors, in MEK, in downstream effectors, KRAS is still there. And in fact, amplification is one of the most common genetic alterations that account for drug resistance. Okay, so much for lung. Now we move into pancreatic cancer. Well, pancreatic cancer, first of all, the G12C mutation is very infrequent, so it was not easy to identify all these patients. But as you can see, the response is even worse than in lung. In fact, we compare the progression-free survival and the overall survival with sotorasib versus classical chemotherapy treatments. You can see it's basically the same, just a little bit, increase, maybe no more than a month. And then the overall survival is basically identical. So clearly, again, the advantage of sotorasib would be only the lower, secondary toxicities, but not certainly in terms of in tumor treatment, in tumor regressions. Okay, so considering that this mutation is very infrequent, the impact of sotorasib or adagrasib in pancreatic cancer is minimal. Okay. Obviously, pharma companies keep making inhibitors. This is just from a recent review published also by Andy Aguirre. And you can imagine all this amount of money that's been dedicated to generate additional, inhibitors of the G12C mutation. Just occupy the entire screen, and then mutations of other mutations like the G12D, which is the most frequent. So at least in this case, not only for lung, but also for pancreatic cancer. And then at the bottom, you can see now the series of pan-KRAS inhibitors and pan-RAS inhibitors at the very bottom. What I'm going to do now is to talk about this one. To talk about this one is RMC 6236 generated by Revolution Medicines, okay? And this is what is going to be the rest of my presentation. This is the structure. On the right is a tool compound that has been used by Ken Olive mainly, so they don't use the compound that is ongoing clinical trials. We had access to it by thanks to a company. I don't know if you ever heard of it. As Medgame, and this would be fantastic for us because obviously we didn't have to depend on Revolution Medicines to give us their compound with all the limitations. We just, get the compound from your company, and this has been a fantastic help in our work. Okay, so this is RMC 6236. Just very briefly, because this is an incredible work by Pirolito. They invented a different method to identify RAS inhibitors using a derivative of cyclophilin A, a molecule that binds certain natural products. So they modify cyclophilin A, so they will bind a series of RAS inhibitors synthesized obviously through medicinal chemistry, and they form binary complex between this modified cyclophilin A and the inhibitors. And then once they had this binary complex, they added KRAS protein. In that particular case, the first case was also G12C, but in this case was in the on stage because they loaded it with a non-hydrolyzable GTP, so the protein remains at least for certain amount of time in the on structure, in the on conformation. Yeah, so they form a three complex between cyclophilin A, KRAS, and the inhibitor, and that is the way they selected very potent inhibitors. This is the mechanism or the method they use to identify 6236. In that case, was the RAS protein was neither KRAS nor the G12C mutation, what they used to form these three complex, and this is the way they isolated the 6236. Now, this is data from the company. It's not, as far as I know, it's not been published, but this is fantastic, okay? This is the progression-free survival, whereas with chemotherapy is, as you saw before, around 3 months. Now, with this inhibitor, even in secondary line metastatic PDAC, they achieve an 8-month progression-free survival. They more than double the results obtained with chemotherapy. And when it comes to overall survival, which is what is really important, there is also a fantastic increase. It doubles. Whereas you can see here the overall survival with sotorasib or with the classical chemotherapy treatments around less than 7 months. Now with the 6236, now they reach 14 months on the average. So this is really doubling the survival. So clearly 6236 now called daraxonrasib. I always use the number, easier for me than to pronounce daraxonrasib. Will have a major impact in the clinic since it improves PFS and overall survival, and obviously has fewer toxicity than standard chemotherapy. So now this company, is my understanding, they already going through phase 3, and I assume that this inhibitor will be approved by the FDA next year, or certainly as soon as possible, because even if 7 months may not sound like a lot of time, obviously for people that are suffering from this terrible disease, doubling their survival uh, on average is certainly a major, a major goal and a major improvement. However, okay, there is always a however, in spite of this obvious advantages of this inhibitor, at least a single agent is unlikely that will increase long-term overall survival in pancreatic cancer, mainly again due to the rapid appearance of tumor resistance. As far as I know, with my couple of clinical oncologists that I can talk to, they unfortunately they tell me that nobody has really gone beyond certainly not reach a 5-year survival. So clearly the solution to this is combinatorial therapies. So here we come. Okay, everything has been kind of, if not introduction, but to give you an overall view of what is going on in the field. Now I'm going to start. I'm going to talk about our work. The way we try to tackle this problem is to hypothesize that we should target KRAS signaling. Remember, this is something we started before it was there were KRAS inhibitors. Okay, this have to put in context with this was about 10 years ago. To target KRAS at multiple levels, including either upstream, downstream, or even in orthogonal pathways, thinking two things. First of all, that maybe targeting 3 targets will increase tumor responses, but especially will prevent the appearance of tumor resistance, because tumor resistance usually is one genetic change. If we target in 3, it's like if you attack the enemy army in 3 places, it's more likely you're going to win than you just only attack in 1 place. So this was our general strategy, and obviously since we don't have patients, and also obviously you cannot go into clinical trials before validating your approach in animal models, we use the mouse tumor models developed by or a variant tumor model, the one that Dave Tubison published in 2003, that carry a G12V mutation in KRAS and P53 null mutations. And with this, what we use these tumor models so to validate our approach, first using genetics, and second to use inhibitors, pharmacological approaches. So let me first tell you a little bit of the model. Again, this was developed by, the basic concept by Dave. What we published now more than 10 years ago is that although the name of the tumor is ductal adenocarcinoma, the cell of origin is an acinar cell. Acinar cells undergo changes between an epithelial and a mesenchymal state, a ductal-like state, something totally physiological and normal. We call it metaplasia. This is normal, happens in liver, happens in kidney, and is absolutely normal. However, if a KRAS mutation occurs, now this reversible stages no longer reversible, so metaplasia is inhibited, and now with the accumulation of other mutations, a series of pre-neoplastic changes occur until finally PDAC develops. Okay, so this is a consequence of the appearance of additional mutations, all of them in tumor suppressors, which obviously, unfortunately, makes this more difficult to target pharmacologically, but that's the way it is. So what Dave did and we did was to use a tumor model based on KRAS mutation and P53. In their model, they activated mutations very early in embryonic development. We do it in a reversible fashion in late embryonic development, but this is irrelevant for what I'm going to tell you today. But what is important is you take a look at this, what I'm going to show you now. I think you don't have to be a pathologist to tell that these lesions are indistinguishable from those occurring in human tumors, including the formation of the tumor, which, as you can see in dark brown, the tumor cells surrounded by a big stroma that actually isolates the tumor from the rest of the organ. Okay, so these models is what we have used in our work, okay? So first, a genetic experiment. So what we use here is an orthotopic model because actually we cannot eliminate a STAT3 from an adult mouse because it's toxic. So we use an orthotopic model. So this is our model. We activate KRAS and eliminate p53 using a yeast recombinase, a flippase, okay? So we activate the oncogene and eliminate p53. We wait 5 to 6 months. Then we add what we call therapeutic alleles, the alleles that we want to see whether they actually have anti-tumor activity. And what we do is to put the mice under a tamoxifen diet and get ablation of the selective nodes. What we did originally was to use two targets. At that time, we only had two. One was CRAF because it's, tumor regression in lung. In pancreas didn't do anything, but since it worked in lung with the same mutations, we thought, well, this could be a good target for pancreas as well. Even by itself, didn't do anything. And the other was the EGFR, the EGFR receptor, because elimination of the EGFR receptor inhibits metaplasia, and as a consequence, inhibits initiation of pancreatic tumors. Not once the tumor is on, then eliminating EGFR receptor doesn't do anything. But if you have it before you induce the mutations, then the tumor does not develop. It's complete inhibition. So either, each of them had some pedigree that could be important in targeting pancreatic cancer. So then we use them, obviously, in this mouse strain that now you can see, and then we put the mice under a tamoxifen diet to ablate RAF1 and EGFR. This was published 6 years ago in Cancer Cell, and the summary of the paper was that we used gene ablation with two targets, one downstream and the other one upstream, and we got 50% of complete regressions. This was the first description of a complete regression of a pancreatic tumors in this kind of models. However, this only happened in very small tumors by mouse standards, and the remaining of the tumor were resistant. Now, later, we amplify these results by testing this hypothesis in large tumors, and unfortunately, we failed. All tumors were resistant to RAF1 and EGFR ablation. So clearly, eliminating these two targets had no relevant consequences with pancreatic cancer. So we started to look for more targets, okay? And we did a lot of omics, as everybody does, but the key experiment came in a simple Western blot. Here you see the resistant tumors, and clearly they are resistant. There's neither RAF or EGFR are expressed, and we saw a direct correlation with phosphorylation of STAT3. Not expression, but phosphorylation in tyrosine 705, which is the residue that is supposed to activate STAT3. So we did a quick and dirty shRNA experiment, and as you can see here, if you inhibit STAT3 within shRNA, nothing happens. The cells keep growing. If now you eliminate RAF1 and EGFR, nothing happens. The cells keep growing, except that now in this scenario, if you eliminate STAT3, all the cells die. Okay, so based on this, we took the time and the effort to go in vivo. So then we use these orthotopic models, now using the STAT3 alleles provided by Dr. Valeria Poli. The system is the same, except here you expand the tumor cells in culture, and then you implant them orthotopically in the pancreas of wild-type mice. You wait for the tumors to develop, and then now in these tumors, we have the conditional three alleles of RAF1, EGFR, and STAT3, and we activate the recombinase, and we see what happens when the recombinase ablates these three genes. And this is what happens. If the mice are not treated, they are all dead in 50 days. However, if we treat with tamoxifen, at 100 days, no mouse is dead. Okay. Now we even switched the diet now, no longer to activate the recombinase. Supposedly recombinase no longer needed, and we were correct. Using the regular diet, the mice were still alive. Okay. And these two mice that died actually didn't die of tumor. They die of other causes. And this is 300 days. This is half-life. The half of the expected lifespan of a mouse. So this, clearly, and then when we sacrifice mice at the end of the experiment, we couldn't find any remnants of tumor or stroma. Okay, so this clearly led us to conclude that eliminating, our original hypothesis was correct, that ablating three independent signaling nodes will lead to complete and permanent elimination of pancreatic tumors. Now, these results have a small caveat, and is that genetic strategies cannot be applied in the clinic. So clearly, we had to translate this approach to a pharmacological system. So we replace RAF1 inhibition or RAF1 ablation, I should say, because first of all, there are no suitable inhibitors against RAF1. And also at that time, we already learned about KRAS inhibitors. So obviously, no matter how effective ablating RAF1 was, we thought that targeting KRAS probably would even be better or much better. So that's when we selected 6236 from your company. Okay. So EGF ablation was replaced by afatinib, which is an inhibitor of tyrosine kinase inhibitor of the receptor family, not only inhibits EGFR, but also HER2. And then this was the real big change. To eliminate the STAT3, we used a selective product developed by Xiaomeng Wang in Michigan, using a very active and very selective product. Okay, so this is the same experiment I showed you before. I will just go a little bit fast here. You have now non-treated or animals treated with vehicle. They grow very fast. Now we always start now with tumors larger than 100 cubic millimeters. So obviously the mice die in about 22 days. All mice die 22 days. If now we treat with RMC, with the 6236, the mice live longer. But interestingly, they double survival. You might remember the clinical trials. They also double survival. Okay, but unfortunately, as it happened in human patients, all the mice die at 45 days, double, 22 to 45, almost exact doubling. Now, what happens if we eliminate the three targets? Or I should say, sorry, we use the three inhibitors, RMC, afatinib, and SD36. The mice survive for 250 days, which is when we we finished the experiment. So clearly, we were able, to my surprise, we were able to reproduce the genetic data with inhibitors, which is really fantastic. We still don't understand the mechanism by which the STAT3, this triple combination eliminates survival. We don't know the mechanism. Okay, this is something we are working on, but it does. Okay, and then you need the three compounds. If you only use two of them, you do not get tumor regressions. Okay, you slow down tumor growth, but you don't get tumor regressions, and obviously resistance appears. Okay, so this is pretty much what I said. Now, this, and also obviously very important, this triple therapy was well tolerated by normal C57 black mice. Now we went finally to the most stringent tumor model, the genetically engineered tumors. Again, with the vehicle, the tumors grow very fast. Now, with the RMC, the tumors grow much slower. Some of them, they can even consider stable disease. And the result is the same that we saw in the orthotopic model. As you can see here in a waterfall plot, 4 out of 5 tumors, we could even think, or even the 5, that they are almost a stable disease. They don't grow very much, and the animals, again, double survival. They go from, again, 22 days to 45, 46 days. Now, there's a paper last year from the Ken Olive group and the Revolution Medicines group, in which the results were different. In using the tool compound, they saw tumor regressions, okay? But to my surprise, and this is something I would like to discuss someday with Ken, because the overall survival result was identical to ours. I mean, double. Without treatment, they live 40 days. With treatment, they live 80 days. So I don't understand why this is such a, important tumor regression. The survival is not any better. But anyway, this is what they published. So now we go to the triple therapy. Okay, since this is what I showed you before, this is with the triple therapy. Okay, so you can see clearly here that there is no tumor resistance. The tumors don't grow back. Some of them don't disappear completely, but they don't grow back. Probably you see it in a waterfall plot. It's more clear. At 20 days, then the mice treated with the 6236, again, 3 out of the 5 tumors can say stable disease. One was really growing, but all the mice treated with the triple therapy, they regress, and by 80 days, only one mouse survived, treated with the 6236, and here that you can see some mice die. The genetically engineered tumors, they usually, the mice are not as healthy as with the orthotopic tumors, but you can see all of them have decreased size. None of them have grown back, okay? Okay, so this is actually, I'm not going to go on, and then again, it's well tolerated, but this is something that anybody is interested in reading. This is the what happened to each mouse, the 12 mice, and just to call your attention here, the 2 mice did not regress completely. Clearly, they went down from 130 to 38 and from 180 to 48, but they didn't disappear. But they didn't grow back, okay? So we don't know exactly why. And there probably is some, other mutation might be coming up. The tumor becomes less likely to respond, but at least for the time, 120 days or 80 days, they did not grow back, okay? So just to finish, I'm going to just tell you the results with human tumors. Human tumors, we use PDX tumor models. We use 5 human tumors. one of them, two, that the one was grown in culture, the other was grown in the animal. And as you can see here, they all have human mutations. Some are the D mutation, the Spartic, the other mutation in codon 61. So there are different mutations. They all have mutations in P53. All of them are point mutations. They are not null mutations. And then some have mutations in SMAD4 and P16, P19, and the receptor for TGF-beta. Okay, so even though having more mutations, all of them responded completely, with the possible exception of the tumors from one of these, from PDX5, which actually has two additional mutation, has lost P16, P19, and has a mutation, sorry, in the receptor for TGF-beta. But in spite of having more mutations, they all respond, or most of them, the majority, and respond very well in spite of being of human origin. Okay, clearly this not, I mean, human tumor cells is not the same as human tumors, obviously, in the patient. But anyway, this is I think it's a very positive result to let us, move further. So, well, this is just a consequence of this experiment. A lot of people, talk about the tumor microenvironment, the relevance of the T cells, to remind people that these experiments are done in immunocompromised mice, so therefore the tumor regressions cannot be attributed to the tumor microenvironment, at least to the immune tumor microenvironment, because there's no T cells around. Okay, so that obviously give us even more ammunition to when we go to treat, whoever goes into treating human patients, they can use cytotoxic T cells to increase the therapeutic efficacy. Okay, so now just the last slide. Clearly, this still is not ready for prime time. There's a lot of work ahead of us. For instance, the SD36 product doesn't have sufficient pharmacokinetic properties to be used in clinical trials. There is now another STAT3 product from a company called Chimera that is already in clinical trial, not for pancreatic cancer, but is already in the clinic. So that's a good positive sign that this compound could have clinical activity, even not because this is immunotherapy, not in pancreatic cancer, but after all, it's in human patients. And the other problem that our clinical colleagues have warned us is that both afatinib and 6236 have they induce toxicity in terms of dermatological rash. Okay, so they think that maybe the combination of both could be a clinical problem or clinical limitation. So here one would be to get improved RAS inhibitors. In fact, I've been told that one of them, which I will for us there's no point really treating using it in mice because mice don't have any toxicity, but anyway, apparently it has by far fewer toxic events in humans. I don't remember now the number, but it's from Revolution Medicine as well. And clearly we are going to try to replace afatinib with EGF products that we already ordered from your company, as well as from antibody drug conjugates, which I think we are also, we haven't yet, but we will soon order from Medchem. So with this is kind of the obvious next steps. And we also need to expand because this is all tested with the V mutation, well, except in humans, it's the D mutation. But anyway, in other combinations, and certainly to try to understand in those cases in which we have identified some tumors that didn't respond to try to understand why. There've been a few cases, but we already are accumulating cases in which some, occasional mice do not respond. So clearly, this is not going to be the only therapy. It's not going to be a magic therapy, but I think this is the first time that a combination therapy has eliminated, at least under experimental models, tumor resistance. So with this, I will finish by introducing the people that work in the lab. In red are all the people working in pancreatic cancer, and I make a special thanks to my long-term collaborator, more than 25 years, Carmen Guerra. She supervised the work. And also in blue is Vicky Liaki, a very talented postdoctoral fellow now that unfortunately for us is going to be leaving the lab. We are trying to do our best to keep her, but she seems to be, and I think it's important also that young people, look for, different horizons in their career. So I wish her the best in her next step in becoming a good, a very solid scientist. So and then our collaborators, just I put in red, a surgeon that he has been absolutely fantastic in giving us tumors, because in Spain, regrettably, most doctors, surgeons do not care about providing samples. And they don't think it's their duty to do that. So anyway, this surgeon has been fantastic, and I put him as our main collaborator. This is the source of our funding, and I would like to thank you for your patience listening to this webinar. Thank you.
Okay. Thanks a lot, Professor Barbacid. Now let's have a look at the questions. Yes, we have one from Joshua Muner. Have you validated the triple therapy approach also in metastatic PDAC setting?
Yeah, very good question. The short answer is no, not yet, because as you know, getting metastatic samples is complicated. A few years ago, they were not even operated. But now, Dr. Sanchez Buenos already provide us with, I think, 6 or 7 samples. And also, we are collaborating with a group in Marseille that they have metastatic samples. So this is, I would say our next, something to do tomorrow. I mean, very, very, very important. And it's something we're going to do as soon as possible.
Okay, great. I think Carl's heading wish to talk. Maybe we can. Hello, Carl.
Oh, Carl Eric. Yeah.
Hello, Mariano.
My good friend. How are you? Nice
Excellent
to hear you.
presentation, Mariano, and very impressive progress. My question is, which is the signaling pathway that leads to STAT3 activation in the pancreatic cancer?
I mean, I'm afraid this question session is not going to be very productive, because the answer is also we don't know. We really obviously know RAF1 because it's downstream, EGFR is upstream, but STAT3, we only did it because we saw this particular Western blot. Otherwise, I would not have thought about the STAT3. It's involved, as you know, probably much better than I do, but in terms of inflammation, etc. In fact, if you eliminate STAT3, inflammation comes up and kills the mice. So, but I just could not relate to the tumor resistance. So this is one of the future projects in the lab, but obviously in the metastatic, the previous question, we know what we're going to do, but in the terms of identifying what is the mechanism, really, if you have any ideas, I'd be more than delighted to hear them.
Right. Well, there are several pathways that are implicated in STAT3 activation, so I guess there is a lot to investigate.
Right. I mean, that's
Excellent.
the problem with transcription factors. They do too many things.
Yeah. Thank you.
PDF receptors are better, right?
Yeah.
Okay, maybe we can continue the discussion with STAT3 from Professor Henning Warzak. How do you think STAT3 is activated? He asked.
Well, STAT3 we know is activated. Okay, that's a good question in which finally I can answer. Okay. It's activated by the SAR kinases and specifically by the FIN kinase. Okay. So when we saw that result, we said, well, instead of using a PROTAC against STAT3, let's use an inhibitor of SAR kinases. So since dasatinib is been already tested in clinical trials and approved and everything, we thought that we were going to have like a short pathway to the clinic. Well, we treated with RMC 6236 with afatinib and with dasatinib in vitro and worked fantastic. Eliminated the cells. However, when we went in vivo, the mice die in 48 hours. So we still obviously don't know what is the main reason. We know that they die because problems in the GI, in the intestine. The intestine pretty much melts away and the mice die rapidly. Why? This combination is so toxic that in just a few hours can kill a mouse. I don't know, but clearly, obviously you cannot even think about going to humans. Even human may be different, but obviously you cannot go to humans with this combination using dasatinib. So clearly STAT3 is activated, maybe activated by some other kinases, but it's clearly activated by the FIN kinase, a member of the SAR family.
Okay, great. We have a question from Marianne regarding in vivo injections. It was a unique or repeated, and do you think that this type of combined therapy is still compatible with radiotherapies or chemotherapy that is already used in the PDAC patients?
Well, clearly we haven't had time to obviously radiotherapy in mice. I admit I don't know how to do it. Obviously we could, we could ask, but chemotherapy will be much easier for us. We haven't had time really to combine, our triple therapy with afatinib, with chemotherapy. Giving 4 inhibitors, I think 3 is already a lot. 4 and one of them being toxic. I don't know. I don't think many patients will resist that. But anyway, something that we could certainly try. We have another project in the lab. We are targeting the tumor microenvironment, and we are using gemcitabine. So clearly, it's no problem in combining with gemcitabine. But I think even three drugs. People don't like to do clinical trials with 3 drugs. With 4 would be even more difficult. But anyway, certainly if it's not enough for patients, certainly combine. In fact, Revolution Medicine already has clinical trials ongoing, combining RMC 6236 with gemcitabine, I believe, or at least one of the cytotoxics. So clearly the cytotoxics are always there, ready to help, even though obviously they cost, quite a bit of toxicity.
Okay. We have one question from anonymous attendees. Would you see a better tolerability if these 3 drugs would be administered locally instead of systematically? Any experience in this regard? Yep.
Locally, no, we haven't really tried. It's much more. I mean, it is not easy to give them. The mice are small. to get to the pancreas. I mean, we are now, in fact, although this is not the question, but we are now operating the tumors in order to try to get metastasis. So because one of the reasons why we don't get experimental metastasis, not human, but mice, is because the mice die before the metastasis really can develop. So we are now operating the primary tumor to allow the mice to live longer and hopefully to get metastatic tissue. But having said this is not easy. I mean, obviously we need to practice more. So injecting within the tumor, we haven't really tried. Afatinib and RMC are oral, whereas STAT3 has a bad pharmacological properties, so it has to be given twice by injection. So.
Okay, great. Speak of pushing into clinical studies. Actually, myself, I have questions. You said there'll be more work. Given that if there's say a approved PROTAC for say or inhibitor for STAT3 in near futures, what would you think is more? What else you need to do before you can say push your triple therapy into clinical trial?
Well, we started a small company, very modest, because funding for science in Spain is very, very limited. But we are trying to make our own. Okay, we are working with medicinal chemists with many years of experience in big pharma, and I mean, we already got some, but obviously they need to be improved. And I'm sure that other companies will do it by themselves. I mean, once this is published, it will be published next week. So, and also, as you know, I've been giving webinars and, I don't like to keep secrets. So, I like to share our results, with our colleagues. That they will be, making these inhibitors. As I said, Chimera already has one that is more potent than SD36. In fact, we already tested the KT-333, and other people will, but it's still not oral. Our chemist or the chemist we are collaborating with say that a PROTAC is a very large molecule, and usually a molecule over 1,000 molecular weight is very unlikely to be oral. I just go for what they say. I'm not, I'm not an expert in that, but I mean, people will try. I mean, PROTACs were not really known, almost for the mainstream scientists 10 years ago. So I'm sure there is a lot of room for improvement. Now, PROTACs are not free of resistance because the problem with the with the E3 ligases. But anyway, that's a completely different issue. So anyway, we're just starting, and I'm sure that other companies really much bigger than what we are collaborating with, should be probably starting to have their programs on STAT3 products.
Okay, great. We have a question from San Hank Lee. Is there any KRAS inhibition strategy in early stage cancer before getting resistance?
I'm sorry, I missed the question. Would you mind?
Yeah, is there any KRAS inhibition strategy in early stage cancer before getting resistance?
Well, no, yes and no. I mean, as you saw in the experimental model in lung, if the tumor is very small and disappears before they can become resistant, yes. But clearly, this is not going to happen in patients. Um, tumors are detected when they're already of a certain size. And one result that I want to share with you that is not a good result, I mean, it's not that doesn't favor, is if we treat with RNC-6236 first, and the tumor gets resistant, then we treat with afatinib and STAT3 product, and they don't work anymore. So they have to be treated the three at the same time. Now, whether this is an issue of pharmacology or, we just have one experiment in which, we interrogated that. I mean, can we treat first with one and then with another? No. For what we know, again, with limited, number of experiments, it appears that the three have to be given at the same time. So, again, for your answer, I think if you treat with only a KRAS inhibitor, unless the tumor is very small, chances are that it will become resistant.
Okay, great. And we have two more questions, but I think it's less related to the research. The first one's Read One Bellows asking if the GEM model it's commercially available, or the mouse models.
Yeah.
It's available commercially, or maybe potential collaborations, maybe.
Yeah, I mean, well, the KPC model developed by Tyler and by Dave Tubison is widely available. I mean, I think almost every lab working with pancreatic cancer has them. Ours is a little bit different, but because ours has the advantage of being inducible. In other words, we already published this, that if we turn on KRAS, we activate KRAS and eliminate p53 in adult mice, nothing happens. The mice are resistant to KRAS and p53. Okay, so this was, very surprising, but anyways, published in Cancer Cell, and I don't think many people are aware because actually the main title of the papers was, it didn't indicate that fact. But now, with the work of Laura Wood and other investigators, in which they found benign lesions with KRAS mutations in healthy pancreas, in other words, in pancreas of people that have died of other diseases or have died of accidents. I mean, this is really scary because basically it's like having a bomb inside your belly, waiting to explode. Because why these benign lesions, which are really panins, do not progress to, I mean, maybe p53 is difficult to mutate, or this is still a year old, this discovery, so there's much more to learn. But anyway, we think I'm getting off the question. I mean, a KPC is the standard model, and my advice is to get a KPC from other laboratory. Dave Tubison very generously has given it to anybody, or even buying them from the company in Maine. From I forgot the name now, but everybody knows it. But then, if they want ours obviously will be also glad to provide it. Okay. So, but as I said, the gold standard as a mouse model is the KPC.
Great. Yeah, let's have the final question from Nicholas Damas. Is the role of the CRAF and the RAF1 MAP kinase dependent on the MAP kinase independent in the PDAC, such as in the lung adenocarcinomas? So basically, I think he's asking if it's the CRAF or the RAF1 is MAP kinase dependent or independent.
Yeah. Well, this is published. We published this, I think, 3 or 4 years ago. 4 years ago, no, more. Anyway, it's published in Cancer Cell, where you eliminate RAF1 and the MAP kinase pathway is unaffected. There's no decrease of MEK phosphorylation or ERK phosphorylation, because this is driven by BRAF, not by RAF1. Okay, so eliminating RAF1, what it does is inhibits the the anti-apoptotic activity of this kinase. So this is the main mechanism. So we are obviously trying to combine RAF1 ablation with other targets, the same way we've done in pancreas, but we haven't been as lucky as we were in pancreas, so we don't have as yet combinatorial therapies for lung cancer.
Okay, right. Thank you once again, Professor Barbacid, for such insightful presentation and for sharing your the great work with us today. And to our audience, thank you for your time and your questions. It is your participations that makes those events so great, so valuable. This webinar is a key part of our mission to foster the scientific communications and accelerate discovery. We're committed to continuing this in 2026 by bringing you more leading researchers through our monthly series. So please stay connected for future events. And from all of us at the Medican Express, we wish you a wonderful holiday season and a prosperous New Year's. So it's goodbye for now. Thank you. Bye.
Okay, goodbye.